audio lectures. So in part one, we mentioned this, we're creating these action potentials or we're creating these electrical impulses. Part two, we want to look at how exactly do we generate these impulses. Okay, so to do that, we have to sort of go back to these plasma membranes that we learned in the cellular chapter. And we want to look at one very important thing with these plasma membranes, and it all comes back to that, the internal environment versus the external environment of the cell. Okay, so we're going to talk about this resting membrane potential, and part two kind of has a bunch of definitions that I'm going to kind of point out, which again are really good, just multiple choice questions. Okay, so we come back to this resting membrane potential, and we get this little paragraph here where it says, when inserted into a neuron and its extracellular fluid, a voltmeter records a voltage across the membrane of approximately negative 70 millivolts. And I always am pretty amazed when I kind of read this little sentence here, that we can actually put a voltmeter into the interior part of a cell and the exterior part of a cell and kind of measure the charge differences between the two. So this is a really kind of cool thing that we can do, measure these differences in something that's kind of so small. But basically what they're telling you is that, they can take this voltmeter and they're telling you the charge on the inside versus the outside of a cell is going to be different. And when they measure it, it's going to be about negative 70 millivolts. Okay, so they're telling you that the inside or cytoplasmic side of the membrane, so inside part of the cell, is going to be negatively charged relative to the outside. So the inside of the cell is more negative than the outside. And then we get this other term where we have these little differences, and we're going to look at polarized here in a second, but they're basically telling you that the membrane is said to be polarized, and that's going to be one of the definitions that we want to kind of pay attention to that we'll come back to in a second. So here is our little picture, and here is our little neuron here, and you can see here's my voltmeter, and I measure the inside of the cell and I measure the outside, and you can see in the picture that the inside is more negative, but the difference between the inside and the outside is going to be this negative 70, and that's going to come back into play as we move through the chapter and turn out to be a really, really, really important thing. Okay? So here are some of our definitions. We have these membrane potential definitions. So we said polarized, and we want to say, okay, what does that exactly mean? It means that there is a difference, okay, or an electrical difference across the membrane. So they're basically just saying if something is polarized, there's going to be a difference between the inside and the outside. Okay, the potential, okay, is going to be the actual voltage difference between the 2 points. So that if I give it like an actual value, that's going to be the potential, okay? And then the resting membrane potential is going to be the voltage difference at rest or when there's kind of no activity. Okay, so these are three things that you want to just kind of be real familiar with. So polarized just means that there is an electrical difference between the inside and the outside. Potential, okay, if I look at this membrane potential, it just is telling you the actual difference between 2 points that I'm measuring. And then the resting membrane potential is kind of similar, but it's basically telling you, okay, what's the difference when there's no activity in the nerve? Okay, but these are really good multiple choice questions where they can be a little tricky, so you just want to make sure you're familiar with those terms and you kind of know what the difference between the three of them are. Okay, so resting membrane potential is going to be determined by two factors, and again, we're going to come back to that kind of cellular chapter and kind of look at that membrane, but it's going to be determined by these two things. So it's the differences in ionic composition, and that's going to be kind of inside versus outside, and then we have these differences in the permeability. Okay, so we want to look at those a little closer and kind of understand how we kind of come to this kind of difference between the inside and the outside and how we get to this negative 70. So here's a picture again from our cellular chapter, and I can see my membrane. And again, what I want to remember from this picture is that I have all these channels in here. And we talked about we have leakage channels, we have things that are kind of pumped across the membrane, but this is kind of what they're talking about when we look at these sort of permeability and we look at these ionic concentrations, is that there's a difference between the outside and the inside, and there are certain things that can move really easily and certain things that my cell's going to actively sort of move from one side to the other. So this is what I mean by differences in ionic composition. So there are differences between the ion concentrations in the intracellular and the extracellular fluids, right? So the inside fluid of the cell is going to be different from the outside, okay? That the inside, okay, so we said the inside and outside of the cell have different concentrations of ions, and this is an important part too, is that we're really concentrating on two specific ones, okay, and it's going to be sodium and potassium. So sodium concentration is going to be greater on the outside part of the cell, and potassium concentration is going to be greater on the inside part of the cell, and we're going to try to kind of maintain this, when our nerves are at rest too. Okay. Now we go to the other one, which is differences in membrane permeability. So the resting permeabilities are going to reflect these leakage ion channels. So we had these channels that just kind of leaked, and we also saw that we had our sodium-potassium pump. Okay, and that's again, the potassium ions are going to diffuse out of the cell more easily than sodium ions can diffuse into the cell. So this is telling you that there's certain things that are moving more quickly, and they're going to lead to these kind of differences in these concentrations on either side of the cell. Okay, so this is my sodium potassium pump, and again, this one, not that it's super important at this point, but this one's going to need that ATP. So it's good to know that this one is a active one rather than a passive one. But these things together, so these leakage channels and that sodium potassium pump, is going to help me maintain that difference between the inside and the outside of this negative 70 sort of millivolts. Okay, and that's said because I maintain it at negative 70, they say that we kind of stabilize that resting membrane potential. Okay, so here is our picture of just all the things that contribute to that negative 70. So I have my little channels here, and then I have my sodium potassium pump, and again, all they're trying to do for me is sort of keep those concentrations kind of different. No more negative on the inside, more positive on the outside. So channels and pumps help to maintain the difference of negative 70 millivolts, and again, they're going to help me to stabilize that resting membrane potential or keep it at that negative 70 kind of millivolts. Okay, and then here's a little video that we're not going to watch kind of as we go through, but these are again kind of similar to the muscle ones, where you can kind of just watch another little summary of all the things that we've just kind of talked about. And again, these are ones where once you've studied it, you kind of know it, you can watch the videos, and then again, if you can follow along, it kind of means that you kind of understand each step and you know exactly what's kind of going on. And there'll be a few extra videos in here that can kind of help to kind of summarize things too. Okay, so why is all of this important? And now we get to the more important part of the chapter. But changing resting membrane potential, okay, so neurons use changes in their membrane potential as signals, and again, we come back to what we learned in the really last chapter, and we learned a little bit easier, is that we're using these changes, okay, as signals to receive, integrate, or send information. So this is how your body is kind of monitoring what's kind of going on around it. Okay, so it can't be produced by stimuli, right? So stimuli is going to be anything that kind of changes that environment. So it's going to be anything that alters ion concentration on the two sides of the membrane, or anything that changes the membrane ion permeability. So these are the two things that help us maintain that negative 70. So obviously any sort of stimuli that changes the amount of concentration of something on the inside or the outside is going to make a big difference, and anything that kind of changes the permeability and allows different things to come in or different things to leave more easily is going to change that difference in that charge between the inside and the outside too. Okay, so it's kind of one of those things where if I know what helps me maintain that negative 70, I know that anything that kind of changes those two things is going to make a big difference. And again, my body is using these changes or this stimuli to sort of receive, integrate, or to send information. Okay, so terms describing changes to the membrane potential, and again, these two are really important too. So changes are always relative to the resting membrane potential, and we get two important terms here. We get depolarization, and then we get hyperpolarization. Okay, so depolarization is a decrease in the membrane potential, and what they're basically saying is that the inside of the membrane becomes less negative. Okay, so it becomes more similar to the outside charge. So the difference between the inside and the outside becomes less than that negative 70. So they're getting to be where they're a little closer to being kind of equal. Okay, and this is going to change or increase the probability of a nerve impulse being created. Because what you're going to learn is that as we move through is that we're going to have this little threshold, and as we get that inside closer to that outside, once it hits a certain level or it gets close enough to that outside part, we're going to create this little electrical impulse and kind of send these action potentials. Okay, so depolarization is a really important term. And again, you want to realize that the inside is becoming less negative, it's becoming more similar to the outside, and that difference of negative 70 between the inside and the outside is going to become less. So it might become negative 50, negative 45, negative 35. And again, this is the really important part for the multiple choice, is that this change or this depolarization is going to increase the probability of us producing a nerve impulse. Okay, hyperpolarization is going to be the opposite. So it's going to be an increase in membrane potential. So the inside membrane is going to become even more negative. So it's going to become even more different from the outside. And we're going to get this greater than negative 70 difference. Okay, and this is going to take us further and further away from creating this nerve impulse, or it's going to decrease the probability of us creating a nerve impulse. And we'll come back and we'll look at this term threshold in a minute here too. Okay, so great changes in membrane potential can be produced by two signals. Okay, so we get graded potentials. And again, for graded potentials and for action potentials, you want to remember which one is over short distances and which one is going to be over longer distances. Okay, so graded potentials are incoming signals, but they're operating over short distances, and they're going to use the dendrites because, again, our dendrites are going to be way shorter, and they're going to be received by the dendrites and received by, remember we said the dendrites are bringing these signals into the cell body. Okay, action potentials are the opposite. Again, this is how my cell kind of transmits impulses over long distances or away from the body, but these are going to be those axons. Okay, so action potentials are signals operated over long distances, and again, these ones are going to go along the axons. Graded potentials are going to be short, okay, short distances, and again, these are going to be my dendrites, and these signals kind of coming into the cell body. Okay, so graded potentials, again, short distance, triggered by some change in the neuron's environment, and that's going to be some sort of stimulus. So again, I have all these sensors everywhere, and I'm going to stimulate these sensors, and they're going to create these little graded potentials. Okay, so stimulus can be heat, light, neurotransmitters, etc. Current stimulus dissipates quickly and decays with increasing distance. So again, that goes with these are going to only be for short distances, and they're going to act as signals over very short distances, essential in initiating action potentials. Okay, so it results in opening of gated ions and voltage channels. And again, this is going to be what's going to help me if I get enough of these graded potentials or enough of these stimulus. I'm going to change that difference between the inside and the outside of the cell enough where I'm going to be able to sort of create this action potential. Okay, so graded potentials, again, short distances, and again, this is all these stimuluses that can change that difference between the inside and the outside of the cell. So now we get to my action potential, and again, long distances, no decay. So this is the principal way that neurons send signals over long distances. We get this brief reversal of membrane potential. So that membrane potential is going to change for a short amount of time. This is going to be what most of us think of as this sort of nerve impulse or this electrical impulse. But this is the important part here, is that it can only be generated when adequately stimulated through these graded potentials. So what happens is I have this kind of threshold that I need to cross, and we're going to talk about this in a second, but I have to get enough stimulus and I have to get enough of a change in that membrane potential to sort of create these action potentials. Okay, so the graded potentials are the only way that I can get enough stimulus and get enough change to sort of create these action potentials and send that impulse down the axon. Okay, so now we're going to look at kind of the steps in generating an action potential. And then we get 4 steps. We get 1 is a resting state. Number 2 is the depolarization. Number 3 is going to be the repolarization. And then number 4 is going to be this hyperpolarization. We're going to get to see this graph here in a second. We're going to talk about each one of these steps and kind of what's exactly sort of going on. And again, generating an action potential just means that we're creating this electrical impulse. This is my brain trying to send information out to my muscle or to one of my organs, or it's one of my receptors sending this information kind of back to my spinal cord and up to my brain to be sort of analyzed. Okay, so here is my action potential little graph. And on here, I can see this little line that says threshold here. We're going to come back to that in a second. But I get this little generation of this little action potential. And I just want to know sort of what each of these little phases and kind of what's going on in each one of these things. So here is the first one. So this is the resting state. So there's no activity in that neuron. Okay, so my leakage channels are open. Okay, my sodium leakage channels are open, my potassium leakage channels are open. I have my sodium-potassium pump. This is what we talked about before, where we have all these things sort of maintaining this negative 70 millivolts between the inside and the outside part of the cell. Okay, but there has not been any stimulation at this point. So here's our little picture again that we saw before, but these are all my channels, my leakage channels, my sodium-potassium pump, and again, these are just working when my neuron is at rest to maintain this negative 70 millivolts difference between the inside and the outside part of the cell. Okay, so now we get to, this is step 1 again, now we're going to move on to step 2. So now we get to depolarization, and what's going to happen, the main thing that happens in this one is that we're going to open up all these sodium channels. Okay, so the voltage gated sodium channels are going to open. The Na is going to rush into the cell, and obviously if I move a lot of sodium into the cell, I'm going to make it less negative, right? So an influx of positive charge is going to depolarize the membrane. So the interior part of the cell is going to become progressively less negative, okay? So it's becoming less negative, it's becoming closer to the outside part of the cell. So depolarization is going to reach a threshold, it's going to become self-generating where it's going to kind of just happen on its own at this point. But what's going to happen is it's just going to increase the amount of this sodium that's going to kind of rush into the cell, okay? So this is my depolarization. So I had the resting state. Now I get the depolarization where the inside part of the cell, all that sodium's kind of rushing in. The inside part is becoming less negative. It's getting closer and closer to the same charge on the outside. And then in the third part, we get repolarization. Okay, so this is where we're trying to create this impulse and we're depolarizing. Repolarization is basically the reversal of all the things that have just kind of happened. So my sodium channels are going to be inactivated. So I'm going to turn those off. My potassium channels are going to open. My membrane permeability to sodium is going to decline. And what I'm really trying to do with repolarization is I'm trying to return to this negative 70. So again, you really just have to sort of learn the first two parts of it, like what's happening at rest, what's happening with depolarization, and then with repolarization, you want to know that, number 3 is this repolarization, but it's basically the reversal of everything that's kind of happened up until this point. And again, I'm just trying to depolarization, right? I'm trying to make the inside more similar to the outside so that eventually I can create this action potential. With repolarization, I'm trying to do the opposite. I'm trying to reset everything to this little negative 70 millivolts. Okay, so here's our picture of repolarization. And again, this was my resting state. I got everything closer together, and now I'm trying to bring it back to this little resting state levels. So hyperpolarization is where, after your nerve sort of fires or after it gets kind of stimulated, what's going to happen is it's going to hyperpolarize everything, okay? So if resting state is negative 70, what it's going to do is keep all these things open, and it's actually going to go a little further past this sort of negative 70. So it's going to make the inside and the outside even more different for a short amount of time, just so that your nerves can kind of reset and that they don't get stimulated too quickly. Okay, so here is my hyperpolarization. And this one, it's easy to remember that it goes past the negative 70 because when you look at the graph, okay, you can see that this line kind of dips a little bit lower down here. And you can see that in the other picture too. If we go back a couple here, if you go back to this one, you can see that this is my resting state here. And I can see at the hyperpolarization, it's going to dip a little bit even lower, and then it's going to go back to that normal sort of resting state. Okay. So those are the 4 steps. And then the next thing we want to talk about is what we saw also in that picture is this threshold point. Okay, so not all local depolarization events are going to produce action potentials. And again, this is an electrical impulse. Okay, depolarization must reach threshold values if an axon is to fire. And again, it's going to take a bunch of these graded potentials to sort of get me to this threshold value and make it where I'm able to fire this axon. Okay, so we come back to this picture again. And again, I can see my resting state, and I can see here I have this threshold line, okay? And what happens is I need enough stimulation to kind of make the difference change enough to where it gets to this negative 55. And then I'm going to create this little action potential, and I'm going to be able to transmit this electrical signal. Okay, so threshold point is typically reached when the membrane has been depolarized, and again, that means it's becoming less negative, by 15 to 20 millivolts from the resting values. Okay, so a strong stimulus is going to depolarize things quicker. A weaker stimulus, it's going to take longer. And if it's too weak, okay, it's going to dissipate too quickly, and my body's never going to kind of get to that threshold kind of point. So here's another little video that you can watch just to review everything that we just kind of talked about and sort of how we maintain things and how we generate the action potential. So again, go through this once you've kind of read your chapter, once you've gone through the PowerPoint and sort of listened to me sort of talking about everything. And then again, these videos are just a great little review where you can hear it just from another person kind of explaining things. So we get this other principle, which is this all or none phenomenon. So an action potential is known as an all or none phenomenon, another really good multiple choice question. And what they're trying to tell you with this is that it means that it either happens completely or it does not happen at all. And it's going to all come down to this little threshold level. Okay, so once the threshold is reached, the action potential is generated and it spreads whether the stimulus continues or not. So it's basically telling you that once I change the difference between the inside and the outside and I hit that negative 55, I'm going to send this impulse whether I continue to get any sort of stimulus at that point or not. Okay, so if I continue to get stimulus, I'll continue sending these electrical impulses. If at that point the stimulus kind of stops, then we'll stop transmitting these impulses along the nerve. Okay, so here is a picture of we come back to our sort of neuron here, and we have our body, we have our little dendrites, we have our little axon, right? So in the body, action potentials are initiated at one end of the axon, and then again they're conducted towards the axon terminal. So if you look at this picture, it's a great time just to kind of go back and review everything. And I can see I have all these dendrites, and again, these are the ones that are going to be sending these impulses into the body of the neuron. Okay, and if I get enough stimulation through these dendrites and I change, right, at this point, if it's at resting state, the inside of this cell is, different from the outside, and again, we're at that negative 70, but if I get enough input into the body and I open up all those sodium channels and I kind of change what's allowed to move in and out, and I make this less negative and I get it closer to that outside and I hit that threshold of that negative 55, I'm going to generate this impulse that's going to travel sort of down the axon, and again, it's going to go towards the end of that axon and kind of reach these terminals and where all these neurotransmitters are going to be, as we talked about. So again, great picture just to sort of go through the steps and sort of think about, okay, what am I trying to do, right? Dendrites bringing impulses into the body, right? Axon, this is my generation of my action potential, and again, I'm transmitting it along that axon sort of towards those terminals. So now we come to another part and we look at these synapses, and we mentioned these a little bit in the other chapter, but we said that, these nerves and these cells are communicating with each other and they're coming into contact with all these other sort of tissues. So a synapse, okay, and we saw this again in the muscle chapter where we had this sort of neuromuscular junction, but a synapse is a junction between two neurons, okay? It transfers information from either one neuron to another or from a neuron to an effector cell, okay? And here's our little picture of my neuron and my body, but I can see I have all these other things kind of coming into contact, and you can tell from the picture sometimes this one can be a little tricky, but the name is kind of telling you sort of where the synapse and sort of how it's kind of being created. Okay, so I can see that I have some things that are synapsing with the body, right, and I have some things that are synapsing with the dendrites, and I have some things that are synapsing with the actual axon, right? So that's where they get their name from, right? It's an axon synapsing with the body, axon synapsing with the dendrite, or an axon synapsing with another axon. Okay, so sometimes the name kind of gives you all the information that you're kind of looking for. But again, this is how they're sort of coordinating their movement and sort of transmitting these impulses between one another. And here is a picture of an actual synapse, which is just really, 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 really cool. So I would never make you kind of label something like this, but again, it's just a really cool picture to kind of look at and see exactly what's kind of going on in, under the microscope. And here's a picture that looks very, very similar to the muscle chapter, okay, but I have my axon here, right, and I have the end of my axon or my terminal with all my neurotransmitters, but I can see that it's going to interact with the body of another nerve cell, okay, instead of with the muscle. But I get the same sort of little picture, and again, your body's going to kind of do this, various ways kind of throughout the different little systems in the body. But this is a synapse between, again, my nerve, right? And then the body of another nerve. So we get different types of synapses, okay? And we get presynaptic neurons, and these ones are going to transmit impulses towards the synapse. And again, sometimes everything kind of comes down to, in which direction are these impulses kind of traveling. So with this one, the presynaptic neuron, these impulses are traveling towards where they're going to interact with something else. Postsynaptic neuron, okay, is an impulse that's actually moving away from the synapse, okay? So it's already synapsed, and now it's sending that impulse, usually to the body. So at a given synapse, you have presynaptic neurons that are going to send information, and postsynaptic neurons are going to receive info, which is what we just said. Most neurons will function as both pre- and postsynaptic neurons. And then this is another cool little fact here too, is that each neuron can have anywhere from 1,000 to 10,000 axon terminals kind of making synapses at the same time. So there's a lot going on with all these little synapses. Okay, so when you come back to this picture here, again, you can see that I can have a lot of different sort of things going on here. Okay, but if the impulse is coming this way, as it's traveling this way and it hasn't got to the synapse with the body yet, it's presynaptic. And then after it synapses and it starts to move in a different direction, it's going to be a postsynaptic. Okay, and they show you that in kind of this one here a little bit too. We'll see another picture here in a second. Okay, so now we come back to our two types of synapses. So we have chemical synapses, which we saw in the muscle chapter. We're also going to have electrical synapses. The chemical synapses are the ones that I kind of want you to sort of concentrate on because those are the ones that are going to be, again, the most common. Okay, so chemical synapses are the most common. They're specialized to allow the release and reception of chemical messengers. And this, again, we saw this in the muscle chapter. So these are going to be my neurotransmitters, and they're going to be made up of two parts. You have an axon terminal. Okay, this part's a little more review from the muscle chapter. And then we're going to have this neurotransmitter receptor region. Okay, so this is my axon terminal. So we have the axon terminal of a presynaptic neuron contains synaptic vesicles or sacs, and they're going to contain thousands of neurotransmitters. Okay, but this is again exactly what we kind of talked about when we went through that muscle chapter, and we were looking at, hey, we stimulate this nerve and we release that acetylcholine. This is what they're talking about with those neurotransmitter molecules released from that terminal. Acetylcholine would be a great example of that. So now we're going to have a neurotransmitter receptor region, and again we saw this in the muscle chapter because we released our acetylcholine, and then we had this binding sort of on that sarcolemma, and this is going to be that little receptor region. Okay, so receptor region on the postsynaptic neuron, usually located on a dendrite or cell body, and again it's going to determine the response of the neurotransmitter. So this is one way that my body controls what's going on, is it releases these neurotransmitters, but then it also has these little binding sites or these little receptors that are going to bind specific neurotransmitters, and then they're going to sort of know what to do based on what they're kind of binding. So again, here is our little picture. So we have our presynaptic neuron, right? Because it hasn't synapsed yet with the body. It's just sending this impulse. But the terminal's filled up with all those neurotransmitters. When the impulse gets to the end, right, it releases those neurotransmitters. And then I can see on the body of this neuron, I'm going to have all these receptors. And this is what those neurotransmitters are going to kind of bind with. Okay? And then I can also go back and be like, okay, presynaptic, postsynaptic. But again, the most important thing to remember is that we're creating this impulse, but eventually it causes the release of those neurotransmitters. And then we're actually going to turn it back into an electrical impulse. Okay, and you can see from this picture too that they're actually not touching, right? So that's an important part too, is that with here you want to realize that there's a little gap in here. So I release my neurotransmitters, and then they kind of cross this little gap here, and then they bind to those receptors. And that little gap there is going to be called this synaptic cleft. Okay, so although close to each other, presynaptic and postsynaptic membranes are separated by the synaptic cleft. And again, this is preventing the nerve impulse from being transmitted directly. So again, it's just another level of control that my body has. So nerve impulses are instead transmitted via this chemical event or this release of those neurotransmitters. Okay, so electrical signals are converted to chemical signals. These chemical signals can then travel across the synapse, and then what's going to happen is, again, we're going to convert them sort of back into this electrical signal, because again, they're going to be that stimulation that stimulates that body, changes the interior versus the exterior, and depolarizes everything and allows me to sort of transmit that impulse along that axon. Okay, so it's electrical, chemical, electrical, but again, just another step of control where my body can kind of control what's happening to a higher degree. So we just want to mention electrical synapses real fast here, and these are just going to be a more direct way that they can communicate. So electrical synapses between neurons provide a simple means of synchronizing the activity of all interconnected neurons. Okay, so this is really how these neurons are synchronizing their activity between each other. So here's a picture of our chemical one. Again, important to just remember that with chemical, I'm releasing a neurotransmitter. With electrical ones, I'm not releasing the neurotransmitter. I'm just transmitting those impulses directly. Okay, but again, those are just ways that the cells are going to kind of coordinate with each other. So now we get to the last part of this part of the chapter. And again, these are the neurotransmitter effects. And again, we already looked at acetylcholine. So neurotransmitters are the means by which neurons communicate. Okay, so the way they process and send messages to the rest of the body. More than 50 have been identified. And most neurons are going to make 2 or more neurotransmitters, okay? And they can exert several different influences. And again, a lot of this is going to be based on the receptors that they kind of bind to. Okay, and these are the ones that I want you to kind of concentrate on, or that I would ask you sort of on your multiple choice test, is the acetylcholine, right? And you want to just know that it's a neurotransmitter, and you want to know what the main function of it is going to be. And this one we already know because it's very important in our muscular contractions. And some of these you've probably heard of too, but we have dopamine, we have norepinephrine, we have endorphins, and then we have serotonin. So those are going to be some of the most common ones that people are familiar with. Okay, and acetylcholine is excitatory. We already know that it's very, very important for those muscular contractions. Alzheimer's is associated with a lack of acetylcholine in certain regions. So no acetylcholine, no, it's important for muscular contractions. And then obviously if there's a disease or a lack of some sort of neurotransmitter for some particular disease, that's an important thing to kind of highlight and sort of remember too. Dopamine, okay, excitatory or inhibitory depending on the receptor, okay, so it can do both depending on how it's binding. This is our feel-good transmitter, okay, so it's involved in coordinating movement, posture. It says for a lot of these, it'll say release enhanced by amphetamines. And again, this is kind of like recreational drug use. What you're going to find is that the reason people sort of use drugs recreationally, a lot of it comes down to just this reaction of these neurotransmitters in the body. Okay, so for dopamine, the release of dopamine or that feel-good transmitter is going to be enhanced by the use of these amphetamines. Okay, dopamine is deficient in Parkinson's disease, so important thing, and it's going to be increased in schizophrenia. Now we get to norepinephrine, excitatory again or inhibitory based on the receptor, important for alertness, emotions, sleeping, dreaming, and learning. Again, enhanced by amphetamines. Decreased levels of norepinephrine are going to lead to depression. Then we get endorphins, and again, a lot of us have kind of heard of endorphins, or I showed you that picture of my little guy when he broke his arm, and it's endorphins are a really, really interesting kind of thing because my little guy he kind of cries pretty easy when he falls and kind of gets hurt. He'll cry pretty easily. When he snapped his arm like that, he didn't cry at all, and that comes down to this endorphins. When you have a really, really bad injury like that, your body is going to release these endorphins in a way to sort of inhibit that pain and make it to where you can kind of get to safety or get yourself to some sort of help, before it becomes a little more dangerous to your body. Okay, so endorphins, generally inhibitory, natural opiates of the body. They're going to inhibit pain, and then they're mimicking the effects of morphine, heroin, methadone. And there's a lot of different ways when we get into like the exercise kind of chapters where you can stimulate this release of these endorphins. Okay, but exercise is a really, really good way that you can do that a little more naturally. Serotonin, again, most people have kind of heard of serotonin. Mainly inhibitory, okay, plays a role in sleep, appetite, nausea, migraines, headaches. Most people know serotonin for more of the sleeping. Imbalances can lead to depression, aggressiveness, impulsive behavior. Drugs blocking its uptake are going to relieve anxiety and depression, okay? So that's a really cool thing to kind of know. And then activity is blocked by LSD or enhanced by ecstasy. So again, you come into these recreational drugs and you realize that the reason people kind of take them is, again, just to kind of change the actions or the reactions of your body towards these different little neurotransmitters. Okay, so this is a really kind of the tricky part of this unit. Okay, is just creating these sort of impulses. So again, you want to go through this one really just like a couple times and sort of watch those videos, and you can watch this video and sort of listen and sort of work through your chapter. But again, you want to concentrate on just those steps of creating this impulse and sort of the role of each of these structures in sort of creating these impulses. And again, this is the one where you get a bunch of little definitions that are, again, really, really important for your multiple choice tests. Okay, so make sure you understand the depolarization, hyperpolarization, and all those, all the terminology stuff that we talked about in this part of the chapter.